So to proceed with histology. So as we completed gross anatomy, it's development, right? So we know so many cells forms the tissue constituents organs forms systems in our body. So you already know the structure of the cell, right? So what does it comprises of? Cell is made up of cell nucleus, cell organelles, and cell membrane. Nucleus, cytoplasm, cell membrane. Embedded on the cytoplasm we have cell organelles, so many cell organelles, right? Like mitochondria, Golgi apparatus, lysosomes, peroxisomes, uh, centriole. Those are all the cell organelles that are embedded on the cytoplasm. They, so many cells contribute to form tissues. So those tissues contribute to organs. This is general in we know. Proceeding with the epithelium, any lining structure of an organ, either it can be outside or inside. If it is a tubular structure, it lines the inner aspect of the tube. Say for example, esophagus, oral cavity. You, for the skin, the epithelium is outside. Am I right? The epithelium is outside to protect us from UV light and various chemicals where we are exposed. For cornea, the epithelium is outside. For blood vessel, the epithelium is inside. So wherever the function is predominant, the epithelium is lining the lumen of the particular organ. So before going with the histology of particular organ, basic things. Epithelium is the covering, I said. So there are various types of epithelium. First epithelium is simple squamous epithelium. Simple squamous epithelium. Simple squamous epithelium. Simple squamous. If you take any epithelium, all the epithelium rests on basement membrane. This line is called as basement membrane. Squamous epithelium on a cross section will look like this, where the cells are flattened and the nucleus is flattened close to the basement membrane. This epithelium is called a simple squamous epithelium. If it is lining the blood vessel, it is called endothelium. Simple squamous. If it is lining the blood vessel, we call it as endothelium. If it is covering the peritoneum, we call it as meso. Same epithelium, same morphology. Depending upon the place where it lines, the name varies. If it is on the blood vessel, it is called endothelium. If it is on the peritoneum, it is called mesothelium. It is called simple columnar epithelium. See, the cells are resting on basement membrane, right? This is the base. This pink color line is the basement membrane. Basement membrane. All the cells are resting on basement membrane. You see only one cell, one particular cell. Any one cell you see. The length of the cell is larger, the width is smaller. Am I right? And the nucleus is more or less elongated or oval in shape. That represent it as simple columnar epithelium. Simple columnar epithelium. Example, GIT. Whole of the GIT is lined by simple columnar epithelium. Whole of the GIT starts from stomach, ends on rectum. Whole of the tube is fully lined by simple columnar epithelium. Now you see the apex of, so this is the way how we draw a simple columnar epithelium. And this is the apex of the cell. And you get a thickening of the cell membrane at the apex of the cell, that we call it as brush border. Brush border. So simple columnar cells, simple columnar cell with the brush border is seen in gallbladder. Seen in gallbladder. Simple columnar epithelium again. But in between columnar cells, you get some flash-shaped cells called as a goblet cell, which doesn't have an apex. Which doesn't have an apex. It is open. Am I right? It is open. That represent the secretions are to be poured on the lumen. Example, large intestine. Example, large intestine. Everything is simple columnar. But we have modification. This is simple columnar. Stomach. Simple columnar with thickening of the apex of the cell, brush border. Goblet cell, large intestine. Now simple columnar with some hair-like projections over the surface of the apex of the cell called as cilia, which is seen on fallopian tube. Which is seen on fallopian tube. Simple columnar epithelium with cilia is seen in fallopian tube. So on simple columnar we have seen simple, brush border, goblet cell and cilia. So that
How to differentiate brush border and
brush border is just a thickening over the surface from the cell membrane. As such, it is an extension from the cell membrane. If you take cilia, you get air-like projections invading outside. Simple cuboidal. How will you describe a cube? Length and width is uniform in shape, so it is called simple cuboidal epithelium. The nucleus is on the center, which is rounded. For columnar epithelium, the nucleus is oval or elongated. Oval or elongated. For cuboidal, it is more or less same. Example, thyroid follicle. Example, thyroid gland. Stratified squamous keratinized. Stratified means many layers. So it's a section of skin where you can see many layers, right? We'll come to the layers later. You can see many layers. So it is a stratified epithelium. Depending upon the presence of keratin pigment, we determine whether it is a keratinized epithelium or a non-keratinized epithelium. It is keratinized or non-keratinized. Palm of the hand is thick or thin? Thick. So this is a section of thick skin. Section of thick skin. Section of thick skin. This is non-keratinized, where the keratin pigment, here you can see the keratin pigment. Here there is no keratin pigment. Again, you don't worry, there are many layers, right? This is the basement membrane. Next you get one columnar layer cell, polyhedral cell, squamous cell, right? So many layers are present along with the absence of keratin pigment. So it is non-keratinized epithelium seen in esophagus and vagina. Esophagus and vagina. This area alone you have to concentrate. Whenever you read epithelium, what is present above the basement membrane alone you have to concentrate. Here it is again transitional epithelium. You get change of one epithelium to other. Am I right? So there are three types of epithelium present on here. One is, these are all columnar cell. Here irregular cuboidal cell. Above you get an umbrella shaped cells. Umbrella shaped cells. So this is called a transitional epithelium, change of epithelium at various layers, which is otherwise called as urothelium. Urothelium seen on urinary bladder, seen on urinary bladder, seen on urinary bladder. So that completes very briefly about it. One more thing is pseudostratified. It seems to be a single layer of cell, but it is a double layer of the cell called as pseudostratified ciliated columnar. Pseudostratified ciliated columnar epithelium, which is the respiratory epithelium seen on trachea. It seems to be a single layer of cell, but it is not so. It is a double layer cell. So pseudo is false, stratified, and cilia is present. So it is pseudo-stratified ciliated columnar epithelium seen on trachea. Respiratory epithelium is pseudo-stratified ciliated columnar epithelium. So up to this you are clear, right? Regarding epithelium. So it is seen on trachea as well as ductus efferens of vas deferens. Ductus deferens of the vas deferens. Next is cartilage. I think this we finished cartilage already. What is this cartilage? Hyaline cartilage. Why you say this is hyaline? Hmm? Chondrocytes are in groups. Chondrocytes, these cells are called as chondrocytes, and these are in groups. So we call it as that. This we call it as cell nest. This we call it as cell nest. These spaces are called as lacunae. The spaces are called as lacunae, and it consists of an homogeneous matrix. Homogeneous matrix, and the outer covering is the perichondrium. Perichondrium. It is the perichondrium. What is the outer covering of bone? Periosteum. It is periosteum. So location of hyaline cartilage is present on the ends of ball and socket joint. Ball and socket joint. Chondrocytes are, these are all the chondrocytes, which are single. Chondrocytes are single, and these fibers are elastic fibers on the homogeneous matrix, and this is the perichondrium. And this is the perichondrium. So the cells are group or single? Single. So what is the fiber present on it? Elastic cartilage fiber is elastic fiber. Pinna is elastic, right? Cartilage of nose is elastic. Epiglottis is elastic. Am I right? Whenever in high pitch sound you shout, epiglottis go for major tension, right? It is not going for tear. That is called as, that cartilage is epiglottis. Epiglottis. So far we completed epithelium. Simple squamous, columnar, simple cuboidal, stratified squamous keratinized, stratified squamous non-keratinized, transitional, and pseudo-stratified ciliated columnar epithelium. Pseudo-stratified columnar epithelium. Name the cartilage. Hyaline cartilage. This is elastic. One more cartilage is white fibrous cartilage, where the cells are parallel. Cells are parallel. Cells are parallel. Location pubic symphysis. It is pubic symphysis. What is this? Section of bone. Section of bone. What you see here, this is the Havesian system. This is the Havesian system. Havesian system or Havesian canal. And these are the active osteocyte. And this lamella is called as interstitial lamina. This lamina is called as interstitial lamina. What is present on the periphery or the margins is called as circumferential lamina. Circumferential lamina. The active cell is called as osteocyte. Osteocyte is derived from osteoblast. Osteocyte give rise to osteoclast. So blast, site and clast. So with two types of bone, right? A compact bone and spongy bone. Whenever you take a section of any bone, the outer layer is the compact bone and the inner layer where the medullary cavity is present is the spongy point. So density high is present on which bone? Compact or spongy bone? Compact bone. It is present on compact bone. So remember, the spongy bone doesn't have Havesian canal, whereas the hard part or the compact part will be having the Havesian canal. Skeletal muscle have cross striation as cross striations. How many nucleus are present? Normal nucleus எங்க இருக்கணும்? சென்டர்ல. skeletal muscle எங்க இருக்கும்? பெரிஃபெரியில இருக்கும். நார்மலி நியூக்ளியஸ் எத்தனை இருக்கும் ஒரு செல்லுக்கு? சிங்கிளா இருக்கும். skeletal muscle எத்தனை இருக்கும்? நிறைய இருக்கும். மல்டி நியூக்ளியஸ். Peripherally placed multinucleus is the unique part of skeletal muscle. Normally nucleus is center, here the nucleus is outside. Normally nucleus is rounder, it is flattened. Normally only one nucleus, here many nucleus. So three things you should remember: peripheral, flattened, many. Three things. That is the peculiar feature of skeletal muscle. So contraction of skeletal muscle takes place by what are all the contractile proteins that are present on the skeletal muscle? Actin and myosin. So that movement that takes place between the sarcomere of each muscle is called as, name the movement, it is called as sliding theory. It is called sliding theory. So this is regarding skeletal muscle. Next muscle is, what are all the muscles we know? Name the muscles. Skeletal muscle. Next muscle is cardiac muscle. Cardiac muscle, you get a connection of one muscle fiber to the other muscle fiber by means of a disc called as intercalated disc. So what is the significance of intercalated disc? Tight junctions are present, so that what makes it, what would be the function of those tight junction? It makes to work rhythmic and synchronized as a single unit, right? It is not acting separately, right? Each muscle fiber is contracting together, relaxing together. But skeletal muscle, it's not so. If one point is contracting, other part will relax. Antagonistic action takes place on skeletal muscle by means of Z line and I line, whereas on cardiac muscle it is not so. Though it has roundly placed nucleus on the center, each fiber is connected to each other by means of interconnected disc, which makes the heart to function as a single syncytial unit. Single, so it works as a single sheet of muscle. That is a peculiar feature of cardiac muscle. Situation of cardiac muscle. Situation of cardiac muscle. Heart. Situation of cardiac muscle. Heart. Next muscle. Next muscle. Smooth muscle. Next muscle is smooth muscle. So single. So if you take one smooth muscle fiber, it is a spindle shaped muscle fiber, closely packed fusiform cells, nucleus on the center. It is present on, smooth muscle is present on the organs. All the visceras have smooth muscle. GIT have smooth muscle on its lumen. The muscle on the larynx, muscle on trachea, muscle on the wall of bladder, muscle on vas deferens. Those are all smooth muscle. What is present on the larynx is skeletal muscle, but what is present on the trachea and bladder vas differences, smooth muscle. Smooth muscle depending upon, and GIT, whole of the layer of GIT starting from esophagus till the large intestine is smooth muscle. So you'll be having 4 layers for GIT. The innermost layer is the epithelium. The second layer is the submucosa. Third layer is the mucosal layer, and fifth layer is the serosal layer. So there are 4 layers for GIT. From inner to outer, the layers are mucosa, submucosa, muscularis mucosa, and serosa. So you are clear with 3 muscle, right? Skeletal muscle is present on upper limb and lower limb. Cardiac muscle is seen only on upper limb, heart, whereas smooth muscle is seen in organs. Leg structure. Artery. Function of artery is to carry oxygenated blood to and only nutrients, oxygen and nutrients. Layers of blood vessel as tunica intima, tunica adventitia, media, and tunica adventitia. So these are the 3 layers of blood vessel. I already told you the inner lining of the blood vessel is called as endothelium. This layer is called as endothelium. This is called as endothelium. Endothelium means what epithelium? Simple squamous epithelium. So there are 2 types of artery, large artery and medium size artery. Large artery example is aorta. Medium size artery is artery of upper limb and lower limb. If the media is thick, that represent the luminous artery. If the adventitia layer is large, it is called as vein. If the adventitia is large, it is called vein. If the second layer media is thick, it is called as artery. It is called as artery. So to differentiate whether it is an artery or vein, for vein, which layer is larger? Tunica adventitia. The last layer should be larger. To identify artery, it is presence of tunica media. I said two types of artery, large artery and medium size artery. Largest type artery is called as elastic artery and medium size artery is called as muscular artery. Here you get much amount of elastic fibers. Since of only smooth muscle fibers, medium size artery. First you should know how to identify an artery and vein. If the artery medial layer is thick, it is artery. If the adventitial layer is thick, it is called as vein. Now on the vein itself, depending upon the substance what is present on the media, if the elastic fibers are larger, it is called elastic artery. If the smooth muscle is larger, it is called muscular artery. And you should know the example of each. And again, vein is divided into a large vein and medium sized vein. Large vein example SVC and IVC, superior vena cava, and this is the veins of the body wall. See on this picture, this is the first layer, which is the endothelium. This is the second layer. This is the third layer. So which layer is larger? Adventitia is larger. Intima, media, adventitia. Am I right? If the media is larger, which artery? Up to this you are clear, right? We have seen epithelium, its examples. Cartilage, the cell of the cartilage is chondrocyte, embedded on amorphous matrix, and type 2 collagen for hyaline and elastic, type 1 collagen fiber on white fibrous, type 2 for hyaline and elastic, type 1 fiber for type 1 for white fibrous for, and there are three layers for the GI, for the blood vessel, intima, media, and adventitia. So vein is a collapsed one, right? It doesn't have a normal lumen. Am I right? If you take artery, on the center what is present is the lumen. The lumen is uniform lumen. Am I right? On artery the lumen is uniform. What about in vein? It is uniform or not? It is not uniform. So you'll be having a collapsed. So on the adventitia you have smaller blood vessel called as vasa vasorum, which is the blood supply going to nourish the blood to the blood vessel. Blood vessel is the tissue. Am I right? It should survey. It is through vasa vasorum. Vasa vasorum. Vasa vasorum is the blood supply to blood vessel. Vasa. And you should know two types of. So artery enters the organ dividing into arteriole. Arteriole, how it ends? It ends as artery to arteriole, arteriole to capillary, capillary to sinusoid, sinusoid to venules, venules to veins. This is how the vein is formed, right? Wherever the blood vessel ends, it arises as venule, then it comes out as vein. So two types of capillaries you should remember. One is a fenestrated capillary, other one is a sinusoidal capillary. What are fenestrations? Pores. Whenever you get some pores on the surface of the endothelium at the level of the tight junction, it is called as fenestrated capillary. So this is the endothelium. This is the endothelium. So whenever you get some pores which is at the level of the tight junction or the margins, that we call it as fenestrated capillary. And whenever the endothelium or the basal ganglia is discontinuous, that we call it as sinusoidal capillaries. That we call it as sinusoidal capillaries. Sympathetic ganglia. Sympathetic ganglia. What is ganglia? We already saw. Ganglia are collections of cell bodies outside the central nervous system. Outside the central nervous system is called as ganglia. What is present on the course of the so this is unipolar ganglia. The type of the ganglion present here is unipolar. This is spinal ganglia. So whenever you read ganglia, this outer covering is the perineurium. It is the perineurium, and these are the axons what has been collected within the ganglion. This is unipolar ganglion. This is unipolar. Here the cells. This is the unipolar ganglion. Here it is. Sorry, spinal ganglia. It is pseudo unipolar. Pseudo யூனிபோலார். எங்க சூடோ யூனிபோலார் படிச்சோம்? ஹெட் அண்ட் நெக்ல ஒரு கேங்லியான்ல சூடோ யூனிபோலார் இருக்குன்னு சொன்னேன். ஆன் cranial cavity. What is the ganglion on the cranial cavity? Trigeminal ganglion. Trigeminal ganglion is the example of pseudo unipolar ganglion. So we'll be having primary lymph organ and secondary lymph organ, right? What is primary lymph organs? Primary lymph organ, secondary lymph organ. Primary and secondary. Primary are lymph node and spleen. Thymus and bone marrow are secondary. Thymus and bone marrow are secondary. So where we get lymph node? What is the location of lymph node? All the regions we have lymph node draining the lymph vessel. Am I right? So this, can you able to see this rounded mass? This we call it as lymphatic nodule. Lymphatic nodule. You divide the lymph node into an outer cortex and inner medulla. On the cortex, you get the lymphatic nodule, whereas on the medulla, only scattered lymphocytes. Scattered lymphocytes. ஒரே ஒரு டாட் வச்சா அது lymphocyte. நிறைய டாட் மொத்தமா இப்படி ரவுண்டா வச்சா அதுக்கு பேர் lymphatic nodule. Lymphatic nodule. Lymphatic nodule, and you get an outer covering for the lymph node. So you'll be having P lymphocytes and T lymphocytes. So majority 30% of the blood contains B lymphocytes. Remaining 70% is T lymphocytes. Up to this you are clear, right? This is divided into an cortex and medulla. Cortex contains lymphatic nodules, whereas the medulla contains lymphocytes. It contains lymphocytes. என்ன தெரியுது இதுல? நிறைய ப்ளூ கலர் டாட் இருக்கு, ரெட் கலர் டாட் இருக்கு. ரெண்டு தெரியுதா? ரெட் பல்ப் and white pulp. So the function of the white pulp is to for immune response. Can you able to make out two things on this? To identify spleen, you should know what is red pulp and white pulp. Red pulp and white pulp. The cells that are present on the spleen is called as stave cells. It is called as stave cells. Stave cells, white pulp, red pulp. White and red pulp. What is this? If I draw like this, what is this? It is called as lymphatic nodule. Lymphatic nodule. On the lymphatic nodule, sometimes an artery can be present. That is called as eccentric arteriole. This is called eccentric arteriole on red pulp. On the red pulp, it is called eccentric arteriole. Eccentric arteriole. Eccentric arteriole. These are all red pulp. These are all red pulp. These are all white pulp. So to identify spleen, what are the 2 points? Red pulp and white pulp. Name the cell. Stave cell. It is called as stave cells. So remember, red pulp constitutes 75 percentage. Here it is 25 percentage. And you should know the splenic cords of Billroth. Splenic cords of Billroth. Many monocytes that if the red cells are arranged as cords, that we call it as splenic cord. Splenic cords, that we call it as splenic cords. Thymus, you concentrate on one follicle alone. One follicle alone. This is divided into cortex and medulla. Thymus is also divided into cortex and medulla. It is divided into an outer cortex and inner medulla. This is one thymic follicle. This is one thymic follicle. If you take, this is cortex, this is medulla, right? On the cortex, you get aggregation of lymphocytes. Everything is T lymphocytes. B lymphocytes are smaller in number. And this is the medulla, which is skated. And corpuscle is present on the center, called as Haysnell's corpuscle. The name of the corpuscle is Haysnell's corpuscle. Haysnell's corpuscle. Difference between lymph node is, on the lymph node, you see lymphatic nodule is present, right? It is somewhat aggregated. The lymphocytes are aggregated. But if you take thymus, it is not aggregated, densely packed. It is similar to lymph node. What is thymus? What organ it is? It is present for us or not? For us, it is present or not? No, it regress by before puberty. By 11 to 13 years, it loses function. Okay, only the remnants can be seen on in front of the arch of aorta. A fibrous tissue can be seen on us. Thymus is very active only on infants. The T lymphocytes, the organ for the formation and maturation of the lymphocyte for infants, not for us. Okay, for us, we have only the remnants on the superior mediastinum. So it is divided into cortex and medulla. Am I right? On the cortex, lymphocytes are present, but it is not aggregated as of lymphocyte. Here the lymphocytes are round clusters. Collection of lymphocyte as cluster is called as lymphatic follicle, which is present on lymph node, but it is not present on thymus. That is the major difference between lymph node and thymus. Tongue, organ of taste. Am I right? So this epithelium is stratified squamous, keratin or non-keratin? Non-keratinized. It is non-keratinized, and you get some glands here. This gland are called as von Ebner's gland. This gland are called as von Ebner's gland. Von Ebner's gland, and this is the epithelium. I'm telling very important salient points penetrating to the histology. Okay, how to identify a particular slide with some salient 1 or 2 points. Okay. Next is fungiform papilla. So we know the location of the papilla, right? Tongue is divided into an anterior 2/3 and posterior 1/3. So clear? So there are various types of papilla, filiform and fungiform. Now we'll proceed with the acini. This is a serous acini. This is a serous this is serous acini. This is mucus acini. See the shape of the serous acini. The cells are pyramidal. Am I right? Cells are pyramidal. Nucleus is rounded. Center or periphery? Center. Center lumen is smaller or larger? Smaller. If these few features are present, it is what acini? Serous acini. It is called as serous acini. Lumen is larger or smaller? Lumen larger. Nucleus is center or periphery? Periphery. Flattened or rounded? Flattened. Cells are flattened or pyramidal? Flattened. So this acini is mucus acini. I'm going to talk about salivary glands. That's the reason why I'm explaining serous acini and mucus acini. You should know first the difference between serous acini and mucus acini. You are clear with this morphology, serous and mucus acini, right? So, mixed salivary gland is parotid gland. Sorry, submandibular salivary gland, which has both serous acini and mucus acini, both serous and mucus. Example, submandibular salivary gland. What we have described on the previous part, right? Those acini will be present on the mixed salivary gland. Mucus, example is sublingual salivary gland, which contains what acini? It is mucus. So it is mucus acini. You know the morphology of serous acini and mucus acini. That will be asked in MCQ. They'll be giving some features. Nucleus is rounded, pyramidal in shape, lumen is larger. So pick up the odd one of the serous acini. Pick up the or they'll give the features of mucus acini, and you have to describe what type it belongs to. So that morphology is important. Larger lumen, flattened cells, flattened nucleus on the periphery, it is mucus acini. Rounded nucleus on the center, pyramidal cells, smaller lumen, it is serous acini. Serous acini, serous salivary gland, parotid gland. Mucous salivary gland, sublingual salivary gland. Mixed salivary gland is submandibular. We have three major salivary glands. Am I right? Parotid, sublingual, submandibular. Submandibular is example of mixed salivary gland. Parotid is example of serous salivary gland. Sublingual, what is on the floor of the oral cavity, is example of mucous salivary gland. Those acini will be predominant. Mucous salivary gland contain much amount of mucus acini. Serous salivary gland contains much amount of serous acini. Mixed, you will be having equal number of serous acini and mucus acini. Next is thyroid gland. Thyroid gland is made up of follicles. Each one we call it as thyroid follicle, lined by simple cuboidal epithelium. The lining epithelium is called as simple cuboidal epithelium. This one we call it as thyroid follicle. Thyroid follicle, which is lined by simple cuboidal epithelium. Simple cuboidal epithelium. Simple. So the lining epithelium of the thyroid is simple cuboidal epithelium. Cells between, this is one follicle, this is another follicle, this is one follicle, this is another follicle. Am I right? Each one is a follicle, thyroid follicle. The space between the two follicle contains cells called as what cell? Para Parafollicular cell. Parafollicular cells between two follicles. Here the cell is called as parafollicular cell. Parafollicular cell. Lining epithelium is simple cuboidal. Simple cuboidal. So we have seen so far epithelium, bone, cartilage, muscle, blood vessel, artery and vein. Two types of artery. Tunica, intima, media, adventitious, three layers. And lymph node, spleen, thymus. Next is pituitary gland. So what are all the parts of pituitary? Pituitary has an anterior pituitary and posterior pituitary. Anterior pituitary, you will be having cells. Here you will be having nerve fibers. This is called intraglandular cleft. This cleft is called as intraglandular cleft. We know so many hormones are produced from anterior pituitary. Am I right? Posterior pituitary secretes oxytocin and vasopressin. Here you get FSH, LH, SH, growth hormone, everything. So that has been secreted by both basophils and acidophils that are present on the basophils and acidophils. So chromophobe and chromophils that are present on the anterior pituitary. Okay. Morning we saw the development, right? Rachy spouts give rise to which pituitary? Anterior pituitary. Posterior pituitary is from